an enzyme. For microorganisms class, napaka-importante. It's very important that we learn their metabolic functions and especially how to control the enzymes. If you know, you already know that we have so many microbial products, diba? Sino ba sa inyo ang kumakain ng kimchi? Mahilig sa suka, mahilig sa nata de coco, mahilig sa patis, ano pa ba yan? Those are all microbial products. Those are products of microbial metabolism. And sa metabolic functions ng enzyme, ng microorganisms, gumagamit sila ng enzyme. And I'm sure that you already have the idea, especially because I have given you the second the second activity for our module on the enzyme to find a published paper where an enzyme from microorganisms is featured, kung ano yung gamit niya, para saan siya, bakit nila in-extract, and so on and so forth. So, fermentation, yung mga pagkain na kinakain natin, usually karamihan sa kanila, products ng fermentation. So, pinakamaraming gamit talaga doon is enzyme. So, ano nga ba ang metabolism? Do you still remember the definition of metabolism in your lower courses like botany, zoology, sa mga nakapag-cell mall na? No, you still remember metabolism? Diba? Alam niyo yung definition yan eh. Kahit nung high school tayo, memorize natin yan. The sum total of all the chemical processes occurring within the cell. So, kahit prokaryotic, ang microorganisms like bacteria, they still undergo metabolic processes. And here is, you also have this in your modules, no? The biosynthetic and the degradative pathways of metabolism. So, pag sinabi natin class na metabolism, there are two pathways, the degradative or that's catabolism, and the biosynthetic or the building up, and that is anabolism. If you join together catabolism and anabolism, and it's gonna be a two-way process, ang tawag natin doon ay amphibolic pathway. So, tignan natin what happens in each of these processes and how is energy participating, yung energy natin na ATP, participating in the process of metabolism among microorganisms. And typical din ito siya class sa ibang cell. Okay, so let's see. Here, ang una nating titignan is the catabolism. Okay. So, catabolic pathways are having this large energy rich molecules. Ano yung mga large energy rich molecules? Protein, carbohydrate, fats and lipids. Ayan, no? Yung ating mga nucleotides. So, sila yung tinatawag natin na large energy and rich molecules. Pag yung large energy rich molecules na yun, mag-undergo ng degradation. We say degradative pathway. Ang ibang tawag natin doon ay breaking down, no? Parang isa siyang malaking block, isa siyang malaking building, tapos i-break down mo siya. So breaking down, that's catabolism. So that it will result into energy poor products. So ano yung mga energy poor products na yun? Syempre, yun yung building blocks ng ating large energy rich molecules. For example, kapag protein nga, ano nga ang building blocks ng protein? We have the amino acid. So, ano ang building blocks naman ng fats and lipids? Ano ang building blocks ng carbohydrates? O, diba? We have the simple sugars, we have the nucleotides, meron tayong ating mga base pairs, no? Yung ating mga bases. Yan. So, yun ang nangyayari. And, the aerobic respiration or aerobic pathway generates more energy. Remember that, ha? The aerobic generates more energy than the anaerobic pathway. But, if we have a look, tingnan naman natin what happens in the biosynthetic pathways or yung anabolic na pathway. Okay, so here, kasi di ba parang umikot lang yan. Galing dito sa degradative pathway, big molecules, papunta siya sa smaller molecules. Kunyari, protein. And then, napunta siya sa amino acid. So, dito naman sa biosynthetic pathway, mag-start ka sa amino acid. So, simple organic compounds, example, the amino acid. And, dadaan siya ng biosynthetic pathway or the anabolic pathway para mabuo mo uli yung large energy-rich molecules such as proteins, lipids, and carbohydrates. Ayan. So, during the process class, merong release at saka input ng energy. So, for example, dito sa ating degradative pathway, nagkakaroon siya ng input ng energy. Kaya, yung product ng degradative pathway, energy poor na siya. Pero dito naman sa kabila, magkakaroon siya ng gain ng energy dito sa simple organic compounds natin papunta doon sa large energy rich. So, there is the storage of energy in large molecules such as protein, carbohydrates, and lipids. So, that's why nga, di ba, in our simple biology classes, we always say that carbohydrate, for example, is the primary source of energy. Kaya kayo kumakain ng tinapay, kumakain kayo ng rice, ano pa ba yan? Ang kanin mo, ay, kumakain ka ng kanin, ang ulam mo, pansit. Diba? So, lahat yung carbohydrate. So, that's what happens in this. In microorganisms, ganun din ang nangyayari sa kanila. But let's see, how many ATPs are produced in each here, no? So, okay, so this one is our microbial respiration. So, this table shows that ATP yield during prokaryotic aerobic respiration. Do you remember what I said earlier? Aerobic respiration generates more energy than our anaerobic respiration. So, there are different sources in prokaryotic aerobic respiration. You have the glycolysis, yung Krebs cycle, pero hindi ko na yan iisa-isahin yung mga methods na yan. Now, what I would like to show you is ilang energy ang na-generate at each stage. So, for example, sa glycolysis, oxidation of glucose to pyruvic acid. Now, ito, nag-generate siya ng dalawang ATP. The production of 2-NADH, nagja-generate naman siya ng 6 ATPs. And then, preparatory step, itong formation of acetyl coenzyme, no, produces 2-NADH. So, meron naman siyang 6 ATP. Sa Krebs cycle, ano ba ito? Hindi na nakikita yung aking slide. Sa Krebs cycle, meron naman ditong 2 GTP sa production of 6-NADH, 18 ATP. Production of 2 FADH, we have 4 ATPs. Na a total of 38 ATPs. This is only for one aerobic respiration. No, this is only one aerobic respiration. Now, Take note of this, class. In one aerobic respiration, the cell consumes 2 ATPs. 2 ATPs. The total yield ATP is 38 in one aerobic respiration of one glucose molecule. So 38. But the cell consumes 2 ATPs during the process. So how many ATPs will be the net yield? The net yield would be 36 ATP. I think there's one question like that in your post-test, na? So merong general term, ha? You have the total yield ATP in one aerobic respiration is 38 ATPs, but the one aerobic respiration consumes 2 ATPs. That gives only 36 ATPs as the net yield. Okay, so please remember that. Now let's go into anaerobic respirations. Excuse me. So how do organisms grow in the absence of oxygen? By the way, class, I have been saying the aerobic and anaerobic. Anong ba pinagkaiba ng dalawa? When we say aerobic respiration, it means that the cell uses oxygen as final electron acceptor. But in anaerobic, what are they using? Just a minute. No, I cannot see my chat box anymore. Some organisms may use an inorganic compound other than oxygen, such as nitrate or sulfate, as the terminal electron acceptor during the regeneration or production of energy in the form of ATP. Please excuse me, class, ha? Wait lang, kasi yung aking isang monitor na tinitignan ko ng inyong mga chat ay biglang nag-blackout. Just wait. I'm just gonna figure this out. Sorry. Hindi ko alam bakit siya nag-restart.
Ay, nag-restart na.
Wala nga. Ito, siyempre naka-live pa rin ako. Pero ito,
Ayan,
wala ako matignan ng chat kasi I need the chat ng aking mga estudyante. Sige lang, habang pinifigure out yung aking isang device, class, I will continue the discussion, okay? So, again, some microorganisms, they do not use oxygen. In fact, you learned this in our previous module that some microorganisms are obligatory anaerobic. What does that mean? In the presence of oxygen, they will die. Mamamatay sila pag may oxygen. So that's why wala silang kailangan ang kanilang environment ay anaerobic. But as with aerobic organisms, anaerobics or the anaerobes must obtain their energy from from reduction reactions. So however, they can generate energy either through fermentation reactions or anaerobic respiration pathways, which utilizes inorganic electron acceptors such as nitrate and sulfate. So kagaya nitong nandito sa reaction na nandyan, so you have the nitrate plus the hydrogen ion in the presence of the enzyme. Ayan, class. Ganito yung function ng enzyme sa mga metabolic processes ng microorganisms such as nitrate reductase. When you see kasi yung ACE yung dulo, ASE, that is usually name of an enzyme. So, dahil reduction yan, reductase, yung nitrate naging nitrite na lang. And there is the corresponding formation of H2O or water. So, the question is, which process is more efficient? Is it fermentation or respiration? Nasagot ko na yan kanina. Siyempre, respiration. That's in the presence of oxygen. Because more ATPs are produced and more NAD plus is regenerated to cycle back into glycolysis. Okay. Next. So, I have mentioned this fermentation to you before, earlier. So, fermentation is a specific type of heterotrophic metabolism that uses organic carbon instead of oxygen as terminal electron acceptor. But in fact, yung fermentation class can actually occur in both aerobic and anaerobic situations because most of the fermenters are called facultative microorganisms, meaning they can exist both in the presence of oxygen or in the absence of oxygen. But usually, they do not use oxygen as the final electron acceptor. So, the anaerobic catabolic process that makes a limited amount of ATP from glucose without an electron transport chain. And that produces a characteristic end product such as ethyl alcohol or lactic acid. For example, tuba. Sino nang nakainom ng tuba sa inyo? Pag ang tuba, newly harvest, ay naku, masarap yan, matamis. Why? It means sugary siya. There is glucose. With the action of organism, pagdating ng hapon, yung tuba na yan, mapait-pait na yan. Tapos, di ba, pag uminom ng marami niya, nagkakalasingan din. Tapos, maya-maya, may maoy pa. But, it means, class, that the glucose has been already converted to ethyl alcohol. So, there is already the presence of alcohol, kaya siya nakakalasing. Yung lactic acid naman, ito yung kagaya ng mga lactobacillus natin. Kagaya sa akin, nag-try akong gumawa ng sarili kong kimchi. Tuturuan ko kayo, papagawin ko kayo ng kimchi sa microbio as part of fermentation. Try ako ng kimchi. And in 2 days time, fermented na rin siya. And that is because of the presence of different species of lactobacillus producing lactic acid. Kaya kapag kumakain ka nun, parang may konting asim-asim din siya ng konti, right? Okay. So now, if we have a look into respiration versus fermentation, ganito siya. So, sa fermentation, dito sa side na to, let me see. Ito, dito, sorry. Ano ba ito? Hindi pa ako marunong. Ito, lactic acid. And here, ethanol. This produces limited number of ATPs. But dito naman, dito sa kabila, yan, 36 ATP. Tapos, meron din siyang carbon dioxide. Bakit nga ulit 36 instead of 38? Kasi, ayan dito sa taas gumamit siya ng 2 ATPs. Okay, there we go. So, next, I have been mentioning enzyme. Kasi pag walang enzyme, walang metabolic process na mangyayari within the cell of microorganisms. Okay, so here is the enzyme. This is the enzyme component. Ganitong itsura ng enzyme. Ano na nga yung enzyme? In your module, enzymes are those that are serving as catalysts. When you say catalyst, that means they hasten reaction. By the way, class, pag nagpa-define ako, pag sinabing ano yung catalyst, huwag kayo magde-define ng, ma'am, catalyst is the one that fasten the reaction. Dahil fast, making it fast, you don't say fasten. Pag fasten, parang ano yan, sabakay ka ng eroplano, fasten seatbelt. No, it has to be hastened, or you just have to simply say to make the reaction faster. So that's the enzyme. But enzymes are not consumed during the reaction. Hindi rin sila na-alter, hindi rin sila nakukonsume, hindi nababawasan. No? Ganun yung mga enzymes. So very efficient. Ano nga yung enzyme efficiency natin doon sa inyong module? How many catalysis in 1 second? So it's 10,000 to how many? 10,000 to 1 million catalysis per second. So sobrang efficient ng ating enzyme. So here we have our APO enzyme. Anyway, gano'n na lang. Okay, APO enzyme. And then, you also have here the coenzyme, or these are the cofactors, the non-protein portion activators of the enzyme. And here is the binding site of the substrate. Okay, so binding site of coenzyme and binding site of the substrate. So when we have the whole enzyme which is active, we call that the holo enzyme. But enzyme class, before they can produce products out of the reaction with the substrate, there has to be the proper fitting. Yung fit na fit talaga sila. So, have a look at this. Tingnan nyo to. Okay? So, in the beginning, when the enzyme and the substrate would bind, hindi siya properly fit. May gap. Here, may gap pa siya. Ba? But there has to be taken into a transition state where both the substrates must bind fitly or properly so that they will produce the products properly. So, hindi pwede yung may gap. Kailangan properly fit siya. Yan yung tinatawag natin na induced fit for our enzyme and substrate, so they can produce products. Okay. So, ano ang importance ng enzyme? Why is the enzyme so important that microorganisms and other cells cannot go away without it? Okay. Let's see. In this slide, you will see this reaction here is without enzyme. So, you will see here the energy or the reactants in here. Okay, this one here. Sorry. Okay, energy and the reactants. And this one here, ito is the energy hill as activated because there is no enzyme. So, it consumes more energy. Okay? So, here, it consumes more energy siya. And, it still produces products. May energy pa rin siya, nakaka-produce pa rin siya ng products. Kasi nga lang, mataas ang activation energy. So, nagko-consume ka ng energy. But, with enzyme, ito, tingnan nyo, but with enzyme, there it goes. It lowers a lot by how many times the activation energy. So, the cell can conserve energy. Nakaka-conserve siya ng energy. Ayan. So, that's what happens, no? Pero, bigyan ko kayo ng mas maganda pang illustration. Ay, ba yun? Bigyan ko kayo ng mas maganda pang illustration. Ito. Now, what happens without and with enzyme? Okay. Here is my energy source. Paano kung bibigyan ko ng energy source itong reactions natin? Have a look. Yan, yung energy source natin. I will pull that to the low energy and see what happens. Wow! With enzyme, even with low energy, the reaction can proceed. But without enzyme, hindi. What about medium? Pag medium na, ganun pa din. So, without enzyme, hindi talaga siya nakaka-proceed. Pero yung with enzyme, nako, it's just so easy. What about sa high? High energy. That's the only time that without enzyme reaction can proceed. No? Bago lang siya nakakapag-proceed. So, that's how the enzymes are very important in the reaction within the cells. So let's see, what are the different factors affecting the enzyme? So in your module, I put there 3 factors. What are those? Substrate concentration, pH, and temperature. For the substrate concentration, as you continue to add more substrate, the reaction will just continue to be faster until such time that it is now concentrated. It reaches the carrying capacity. So, hindi ka na pwedeng mag-add kasi hindi na kaya ng cell. Pero, you just have to remember that enzyme functions will continue. It will become faster with the addition of substrate only and only until saturation point. Okay, so the thing about temperature naman and pH is a little different. So I will show you the effect of pH. Okay, before I discuss this, you just have to remember that in your first module, meron tayong iba't ibang klase ng microorganisms depending on their pH requirement. Meron tayong tinatawag na acidophile. Di ba pag sinabi nating philic, ibig sabihin nun they are loving, acid-loving, acidophile o acidophilic. Meron din tayong alkalophile, no, yung mga alkaline-loving. But most microorganisms, they thrive in near neutrality. Pero wala tayong tinatawag na neutralophile or something. Wala pa akong na-encounter na ganun ang tawag. We have alkalophile, we have acidophile, pero wala pa akong na-encounter na neutrophile or neutralophile or something. Let's see. So here in this graph, I'll be showing to you 3 cases. So this one, these are microorganisms thriving near neutrality. So, between 6 to 8, yan yung kanilang pinaka-optimum. Ito naman sa mga alkaline, no, basic environment. And this one naman is in the acidic environment. So, depending on the pH requirement of the microorganism, especially like bacteria, they can grow optimally. Itong mga peak na ito ng, ito dito, pag kunyari here, and this one. Ibig sabihin, class, ito yung mga optimum optimum pH nila. Diyan, mas pinaka nag-grow yung either acidophile, alkalophile, and those that thrive best at neutrality. Yan. So, tandaan nyo lang walang, depende sa microorganism, depende sa inyong species, they have different pH requirement, pero ang pinakamarami ay between 4 to 9, where the optimum pH is between 6 to 8. Slightly acidic only and slightly basic, and that is near neutrality. Near neutrality siya. Okay. So what about temperature naman? Ito, sa temperature So, what happens if we apply different temperatures to our enzyme? Ito, nakikita nyo tong nandito sa sa side na to, no? Okay. So, here we have the green, the enzyme here, enzyme. Yung black naman, substrate. Yung blue naman is the product. So, pag dito sa area natin, dito banda sa taas, tingnan ninyo kung gaano karami ang substrate at gaano kadami ang napoproduce na product with the binding of the substrate and the enzyme. Tignan ninyo yung blue. Bantayan ninyo kung gaano siya kadami at each temperature. So, for example, start tayo sa 10. Start tayo sa 10, no? Hindi gumagalaw. Ayan, okay. So, 10. At 10, mabagal ang movement ng substrate. Mabagal din ang movement ng product, ay mabagal ang production ng products with the binding of the substrate and the enzyme. Pero increase natin to 15. Tingnan ninyo, medyo bumilis, no? It's a lot faster now. And what about if we go up to 20? So it's getting faster and faster. As we move up to 25, there we go. We are producing more products now as there are more bindings that's happening between enzyme and the substrate. So 30 degrees centigrade. Mostly, microorganisms class grow best at 35 degrees centigrade. Itong 35 na to. That's how we usually set when we are incubating them. And then this one is at 40. Okay, this one is at 45 and this one is 50. And let me pause. Okay, did you notice something? Na-notice ninyo yung difference ng structure ng ating enzyme? Our enzymes and the enzymes of microorganisms are actually very sensitive to heat. So, they can denature. Yung term, you have to remember, denature the enzyme. So, hot temperatures can denature the enzyme. That's the reason kung bakit yung ating boiling nakakapatay ng microorganism. Yung ating sterilization nakakapatay ng microorganism because their enzymatic activity is disrupted. Nawawala because the enzymes are denatured. So, what happens if we continue? Nasa 50 pa lang tayo, 50. There. So, that's 50. We're still producing a little products. And then, as we move to 55, konting-konting products na lang and our enzymes are more denatured. And this one class is 60. May products pa ba tayo na po-produce sa 60? Wala na. Our enzymes are properly denatured. They cannot bind the substrate anymore. Substrates and the enzymes are not compatible anymore with each other. Hindi na sila nakaka-bind, hindi na nakaka-produce ng product. I'd like you to see this again, alright? And I would like you to notice the details. So, just to show this again to you, we're starting with 10. Molecules are moving very slow, producing a few products, binding with substrate and enzymes also very slow. And then, as we move up, we go to 15. It's starting to become more and more faster. So, more products are produced at 20. And this one is at 25. And 30. Ayan. And we also have 35. So, the optimum. Dito talaga, class, mas marami ang napoproduce ng ating enzyme. Okay, at 40. 45. And 50. That starts with the denaturation of our products. Enzyme. And then 55 and 60. So there. The enzymes are all denatured and no more products are produced. Okay. Did you get that? Okay na yun, naintindihan na. So, what are the different mechanisms by which the enzymes are controlled. Dapat makontrol itong enzyme. The knowledge here that we can generate will help us first control the growth of microorganisms. If not control, allow them to grow. Sometimes we need to culture these microorganisms, especially if the enzymes that they are producing are very beneficial. Okay, for example, of this control is, in your notes, diba, you have allosteric activation. What happens to allosteric activation? Now, before I discuss this, I just have to let you know that an enzyme, just like what you have in here, has two binding sites. One is what we call the active site. This is where the substrate binds. The active site of an enzyme is where the substrate binds. But there is another site. Here is what we call the allosteric site. Allosteric site means the other site. Apart from the active site, there is another site. And this allosteric site is either activated or inhibited. Or in other words, this can activate the enzyme or this can inhibit the enzyme from binding the substrate. So let's see first what happens to this allosteric activation. Have a look. Okay. So when the allosteric activator site is empty, the substrate cannot bind because they are not compatible. Oh, look at that. Because our allosteric site is empty. But the binding of allosteric activator changes the conformation of our active site. So now the substrate can bind. That's how important our allosteric activator. Okay? So the allosteric activator works like that. Let me just repeat that again. So when the allosteric activator site is empty, the substrate cannot bind because they are not compatible. Once the allosteric activation or allosteric activator binds with the enzyme, that changes the conformation of the active site, so the substrate can now bind. So that's allosteric activation. What about allosteric inhibition? So this is different naman. Allosteric inhibition is when the allosteric inhibitor in here is present in the enzyme or present in the allosteric site, the enzyme cannot bind the substrate. They are not compatible. So let's see. No? Alright, so this is a little opposite with the allosteric activator. Here, when the allosteric inhibitor is bound to the allosteric site, the substrate cannot bind. That allosteric inhibitor must be removed so that the conformation of the active site will become compatible with the substrate. So this time, the substrate can bind with the enzyme and produce product. Okay. So, did you get that? Okay. So let me just replay that to you. When allosteric inhibitor is bound to the allosteric site, our substrate cannot bind. This allosteric inhibitor must be removed, and now our substrate can bind as they are now compatible with the active site of the enzyme. So that's how it goes. That's allosteric inhibition. Now there is another control of the enzyme that is very important for us to know, and this is called the feedback inhibition. Mostly the cell works with a negative feedback inhibition. So it can be both. No? It can be positive, it can be negative, but mostly our cells move or functions in a negative feedback inhibition. So what happens to this? I just would like you to watch this first. Okay? So there. Now, let's see. We have here a series of enzymes. The enzyme 1, enzyme 2, and enzyme 3. And they act all together to produce products. So these enzymes are where substrates bind. Na ito yung ating mga itong ating mga purple things na ito. They are substrates na produce into end product. Once the substrate binds with the enzyme, they are converted into end products. But once the end products accumulate, yan no? Wow, diba? Sobrang dami na ng end product na nag-accumulate. Ibig sabihin nun, the cell must signal that the production should stop so that there will be no wasting of resources. So what happens? One of the end products will bind to the first enzyme, or it can actually bind to the second enzyme or the third enzyme, as long as it will bind to any of the enzymes in the series, and that will stop the substrate from binding to the enzyme so that no more end products will be produced. This will help the cell conserve energy, conserve resources. Okay. So now next, what are the importance of enzymes? The importance of microbial enzyme. This is the question that I would like us all to answer based on the paper review that you made. Nakagawa na ba kayo? Okay, so I'm gonna look into in our chat box now. I would like you to have a look. I would like to, acknowledge muna the people who are present. Kanina na hindi ko pa nabasa. Sino bang last ko binasa? Si Maureen. And then we also have Judy Ann, Kim Bernadette, at tapos ko na pala to, si Cirilina, si Anna Joy, Jaisel, Crizel, Dia, Angela, Zofia, Jorilyn, Casey, Donna, France, Richard, Jeremy, Lauren Joy, Janry, Jamaica, Feliciana, Alexil, I have Giselle, Emily, Rosalie, Chevin, Aaron, Chrislyn. Okay. So I would like you now to chat any of your questions regarding our first discussion. Chat na kayo, class. So, what is the importance of microbial enzyme based on your research? Sino na yung mga nakapag-research na, nakagawa na ng kanilang paper digest? Come on, I'd like to hear from you. I'm waiting. Can everybody still hear me? Yung nakakapanood na hindi nakapasok sa YouTube for some reason, you can use your Facebook chat, yung ating GC, to let me know that you are watching, and you can also post your questions in the GC if you are not in our live chat box. Okay, I'm still waiting for your questions in our live. Ongoing pa, hindi nyo pa nagagawa. Sige. That's okay. But, ito, microbial enzymes from Jovan. Microbial enzymes play a major role in the diagnosis, treatment, biochemical investigation, and monitoring of various dreaded diseases. Profound naman masyado. But that is very true. No? Microbial enzymes are very, very important in medicine. Alam nyo yung mga enzymes na yan, once isolated, those enzymes are the ones that we can actually develop into an antibiotic. Yeah? Ano pa yung nasabi niya? Treatments, biochemical investigation. Yung mga biochemical investigation na yan, masyadong general, no? Pero, one example dyan, class, is bioremediation. Bioremediation of polluted area, for example, oil spill, yung ating mga heavy metals. You can actually have microorganisms, either single or consortium of microorganisms, in order to address those things, and that is still via microbial enzyme. Kahit yung ating removal of the dirt talaga, the removal of the toxic parts and the cleaning of the sewage. Yung sa ibang bansa, they recycle water. Yung mga galing sa kusina, galing sa banyo, ganyan, they recycle the water and they treat that, no? That's called the treatment of sewage, or sewage treatment, and they use that again for agricultural purposes, pang dilig ng halaman, pang pakain ng livestock, na pangkain ba? Pangpainom kasi tubig eh. And ginagamitan pa rin yun ng microorganisms to finally remove the contaminants. I'll be preparing another video for you, our tour to the sanitary landfill of Puerto Princesa, and I will be emphasizing the use of microorganisms in how to address problems, especially in sanitary landfill, for example. Okay. Yeah. Sa ano yung sa Japan, Maricar? Oh yeah, that's correct. Kaya po ang linis ng drainage nila. That's correct. Yeah. When I was studying in Japan, na yung aming mga wastewater talaga may pinupuntahan. And halimbawa, pag sa laboratory, may mga toxin yan, mga heavy metal. Iba yung septic tank niyan, iba yung pinupuntahan. Pag sa mga bahay-bahay lang, iba rin ang pinupuntahan. At weekly, merong nag-collect nung water doon sa tank para dalhin doon sa kanilang sewage treatment plant. So, kaya malinis talaga. In fact, totoo yan, Maricar. Sa Japan, even yung water nila habang nagsa-shower ka, pwede mong inumin. Nakakaganon nga ba nagsa-shower? Kasi it's really malinis. Yes, malinis. Okay. Arnel, according to published online research that I've summarized, very good, Arnel. So you've done with this activity already, The microbial enzyme is important for agro-industrial waste by biosurfactant producing microbes such as amylase, protease, and lipase. That is so correct, no? With microorganisms, ang dami mo talagang pwedeng gawin. In fact, ang daming pwedeng magawa in fields of medicine, in fields of food industry, even sa paggawa ng paper, sa paggawa ng mga products na binibenta. Ang dami. Totoo yan, no? Biosurfactant, yung amylase, fructose, and lipase. Very good, very good. From, kanino to? Masiela Mulaga. Kanino ito? Jerry Ann, sa'yo ba to? Ang Mulaga? Masiela? Paano ko babasahin yung pangalan mo? Teka ka. Hanapin ko muna. Anyway. Oo nga, baka ano. Sige, find applications in pharmaceutical and food productions. That's correct. Application in pharmaceutical. Meron akong ipopost na video for you to watch. One of our studies na kinandak namin, yung naka-isolate kami ng bacteria from abalone, yung kinakain natin na sobra-sobra from abalone, that is antibiotic producing. So later on, kayo, kung interesado kayo sa mga ganitong fields, you can actually have this as your thesis. Nakita nyo naman sa pag-feature ko, diba? We already have basic microbiology laboratory na pwede nyong magamit pag nag-thesis na kayo if you would like to do thesis on microorganisms para sa mga major sa bio. And tama nga, si Jerry Ann nga. Okay. And then si Giselle May, microbial enzymes play a major role in food industries because they are more stable than plant in animal and animal enzymes. Correct, no? Food industries, nabanggit ko kanina, we have so many foods na ginagamitan ng microorganisms, di ba? Isang pagkain yung Yakult. Sino bang paborito ang Yakult? Ako, paborito ko ang Yakult. I wanna have one Yakult every day. That's microorganism. If you look at it under microscope, naku, makikita mo. It's really moving. Sayang nga lang wala tayong laboratory. I was so excited pa naman sana to show these things to you in the lab. Pero who knows, baka pwede na tayo next year, no? From Christine, in detergent industry, they are used as additives to remove starch-based stains. In paper industry, they are used for the reduction of starch viscosity for appropriate coating of paper. Oh, very good, Christine. You've done your homework. Now, it's really true. Alam nyo, hindi simpleng sabon lang yung mga sabon na ginagamit natin sa kusina. Yung panlaban natin. Microorganisms are still used to completely have a refined detergent. And that is to remove the starch-based stains. Ayan. So, nakita nyo ba yung iba ng mga sabon natin? Kapag minsan ginagamit nyo siya sa mga puting damit, parang nag-stain siya. O, ibig sabihin, hindi properly removed ang starch-based stains nila. So, sa paper industry, they are also using it to reduce the starch viscosity para maganda yung quality ng paper. Glossy ang surface and pwede siyang maibenta ng mas mahal kaysa doon sa simpleng papel lang talaga siya and you cannot even use it para maging mas maganda ang output niya when you print on it. Okay, any other comments? Any question? Okay, so this is for my microbiology class. Sunod dito, I'll be discussing very briefly, but please microbiology class, stay in tune na, hindi pa tayo tapos. Mga another 30 minutes na lang naman for the cell cycle. Any question regarding your module? Babalikan ko kayo. Maglagay kayo ng question dyan sa ating live chat regarding the module. Kahit pa yung question class ay galing sa inyong post-test. If there are parts of the post-test you do not understand, please ask me and I will answer it here. This is your very good chance na to perfect your post-test. Okay, so now I'm going to share naman yung aking slides sa cell cycle. Don't go away, my microbiology class. You can still relate to this. Mabilis na mabilis lang ito. Okay. Can you see now? Can you see my slide? My cell cycle slide? Let me just check dito sa aking isang device. Meron ba akong mga na-miss na question? Okay, very good. Nakikita. Okay, so cell cycle, mabilis lang to. Stay in tune, cell mold students. Okay, so the cell cycle class, when we say cell cycle, talagang nasa nucleus yan, no? We have here the details of nucleus with the three regions, the nuclear envelope, the condensed chromatin, and the nucleolus. And in the nucleus class, this is where we can find our DNA materials enclosed in the chromosome. So ito yung ating chromosome here. Chromosome composed of two sister chromatids, dalawang sister chromatids, exact copy of genetic material in each chromatid. And at the middle, we have the centromere. So meron tayong tinatawag na P-arm, Q-arm, and the telomeres. Yan. So, I will not go into the details of the DNA yet as we will have a separate lecture for this. Okay. Separate lecture tayo, no? And what is the structure talaga of the chromosome? Okay, I would like to show you the details of the chromosome. Ano ang itsura ng chromosome? Okay, so sister chromatids ito of a metaphase. So at metaphase, the chromosomes are in a duplicated state and are at their most condensed. So very condensed siya. Kasi in the beginning, chromatin thread lang yan eh. Chromatin thread, meaning manipis siya. Pero pag nag-condense, meron na tayong chromosome. When chromosome is condensed, interaction among chromosomal proteins keep loops of the DNA tightly packed in a supercoiled array, just like what is shown. That's super coiled talaga siya, kaya nagiging condensed ang chromatin thread into chromosome. On a closer examination, we can see that the chromosomal proteins and DNA are organized as a cylindrical fiber. So if we immerse a chromosome in salt water, this fiber loosens up to reveal a structure like this. Naalala nyo yung ginawa ninyong experiment sa yung extraction of DNA, yun ang dahilan, no? The string is one molecule of DNA. Each bead is a nucleosome that consists of a DNA molecule looped around a core of histones. Nucleosomes can loosen in controlled ways. At hindi ko na mabasa ang nasa ibaba. At saan ba yung banda? Ayan. So, you can see here the DNA and the histone. DNA and the histone compose the nucleosome. Ayan. So, that's the details class of the DNA, the chromosome. Yan yung merong sa loob ng chromosome. Nakikita mo yung double helix here, oh. Ano ba yun? Nawala. Again. So, you can see Balik-balik ako. Yan. In a closer examination, we can see here the chromosomal proteins and the DNA organized in cylindrical fiber. Tapos, we will have a closer look. So, yan yung nucleosome. Oops. Okay. Nucleosome. You have the DNA and the histone. Tingnan nyo yung DNA sa loob, oh. You can still see the double helix. That's what it looks like. Okay. So, that's chromosome. Now, chromosomes, because knowing the chromosomes is actually very important. And one of the activities that involves chromosome is actually karyotyping. Karyotyping is very important in the process of knowing the characteristic of an individual, including any abnormality. In fact, even sa womb pa lang yung baby, you can already get a karyotype like this through amniocentesis. Yung amniotic fluid, pwede kang magkuha ng konti nun that has the chromosome of the baby para baby pa lang, malalaman mo na kung yung baby sa tiyan will become abnormal or not, especially if there is vulnerability. What's vulnerability? Yung predisposition. If the family, it runs in the family to have the abnormality. Kaya nga class, you know, in other countries, abortion is legally done. Kasi, pag nalaman nila that the baby inside the womb will become an abnormal individual, ina-abort na lang siya. Diba? So, here is the karyotyping. Have you done this in your activity? Karyotyping is an arrangement of chromosome from the biggest to the smallest. So, ganun kayo dapat mag-a-arrange nun, na biggest to smallest. Tapos, syempre, ang pinakadulo is the X and Y chromosome. X and Y, meaning that's the sex chromosome. If you have XX, that's female. If you have XY, that is male. So, ano ang examples ng mga abnormalities? Examples, class, are here. So, this is the normal male chromosome with an XY chromosome here. Yan o, XY. Normal XY chromosome. So, this one is a male. But, if it becomes abnormal, like for example here, Ayan. The Edwards syndrome. Here, there is trisomy in chromosome number 18. Imbis dalawa lang, nagkaroon siya ng tatlong chromosome sa number 18. So, for human beings, there are actually 46 chromosomes. So, sa kanya, Edwards syndrome, meron siyang 47 trisomy. What about ito? Patau syndrome. Sa kanya naman, merong trisomy in chromosome number 13. So, 47 chromosomes din siya. And then, Klinefelter syndrome. Meron siyang trisomy in the sex chromosome. XXY. Aroy. So, what would be the characteristics of these individuals? Tingnan natin. Let's start with the Edwards syndrome. Pag Edwards syndrome, trisomy number 18, so, usually 80% female siya nangyayari, no? Majority die before birth. Low survival ang rate. Symptoms are having small head, malformed ears, widely spread eyes, clenched hands, heart abnormalities, kidney malfunctions, etc. No, kawawa sila masyado tignan. And then, what about ito, Patau syndrome. This one is Patau syndrome. So, meron siyang small head, wala siyang eyebrows, o parang Mona Lisa lang. Walang eyebrows. Cleft lip, dysplastic, malform ang ear, clenched din ang kanyang hands, may polydactyly pa. Ano yung polydactyly? Kung 5 lang yung fingers natin, pwede maging 6, pwede maging 7, yan ang polydactyly. And undescended or abnormal testes. Yan ang Patau syndrome. What about, Patau. Ito naman siya class ay Klinefelter syndrome. Hindi mo alam kung babae ba to o lalaki. Not taller, taller but less muscular body than males. Broader ang hips niya na karakteristik naman ng female. Larger ang breast na karakteristik ng female. Tapos weaker ang bones, low ang energy level, smaller ang penis and testicles. Delay ang puberty, less facial and body hair following puberty. So, dahil dalawa ang X niya, may Y siya, para siyang male na parang female. No? That is Klinefelter syndrome. And yung Down syndrome na familiar kayo, Down syndrome. Ang Down syndrome class ay trisomy naman sa number 21. Ang number 21 niya na chromosome ay 3. So, Down syndrome or anong tawag din yung dito? Mongolism, mongoloid. O, nakakita na kayo nito. So, typical facial features niya ay ganito, parang mga drooping eyelids, flat ang forehead, brush-filled ang kanyang iris, tapos parang bulging ang eyes, minsan fissured ang tongue niya, meron ding pwede magkaroon ng clinodactyly, yung wide ang gap between first and second toes, short, broad hands. Tapos usually ang lifespan nila hindi masyadong nagtatagal. Yung iba up to mga 15 years and or less. Ayan. So, Down syndrome. Okay, so I will continue straight para mabilis lang tayo to our cell cycle. This is our cell cycle. The cell cycle consists of mitosis and interphase. Usually, the longest of the cell cycle is yung interphase na portion because that comprises the G1, the S, and the G2. Let's look at the G1. Ano nangyayari sa G1? G1 of interphase occurs after daughter cells are formed by mitosis and cytoplasmic division. During G1, cells build most of the proteins, carbohydrates, and lipids that they use or export. Many cell types become arrested in G1. They carry out their specialized functions, but they do not divide. Okay, so that's in G1. In S, tandaan nyo lang, basta S, yung word dyan is synthesis. Now, there is DNA synthesis, chromosome duplication in the S stage after mitosis. So, what happens naman sa G2? G2 is the interval following DNA replication. During this time, the cell produces proteins needed to drive mitosis to completion. Ayan. So, that's our cell cycle. Now, let's go to mitosis. Mitosis is the division portion. So, this divides the cell into daughter cells exactly complementary or similar, the same to the parent nucleus, the parent cell. This is usually followed by cytoplasmic division. So, let's go into the details talaga of mitosis. So, ano ang mga stages ng mitosis? I just would like you to remember PMAT. Ano yung PMAT? Prophase, metaphase, anaphase, and telophase. Pero syempre, in between, minsan meron pa yung mga pro-metaphase, pro-anaphase, and so on. Pero, primarily, 4 lang talaga ang stages ng mitosis in the order of prophase, metaphase, anaphase, and telophase. So, quickly, tignan natin kung ano ang events in each of this. Okay, so, let's start with animal cell. Now, in animal cell, what happens here in early prophase? So, there is the duplication of chromosome so that they will begin to condense. Each chromosome consists of 2 daughter chromatids joined at the centromere. What happened in late prophase? So, chromosomes continue to condense, new microtubules assemble and move one of the centriole pairs to the opposite side of the cell. Okay. So, in prometaphase, during this transition to metaphase, the nuclear envelope breaks. So, wala na yung nuclear envelope. Microtubules penetrate the nuclear area and form the spindle apparatus. Many spindle microtubules are built in here in preparation for the proper alignment. Ayan, metaphase. This is where chromosomes are properly aligned at the equatorial plane. Pag metaphase class, ang tatandaan nyo na keyword is alignment. Alignment of the chromosomes at the equatorial plane. They are most condensed. Pag anaphase naman, ang tatandaan nyo dito is separation. Ano ang nagsiseparate? Sister chromatid, hindi yung chromosome, ha? So, sa middle, nag-align sila, pair sila, isang chromatid pupunta sa opposite pole, ang isang chromatid pupunta sa opposite pole. Okay, sa telophase, magkakaroon na ng dalawang daughter cells. And it will continue to interphase. So, interphase, yung naging single-stranded na sister chromatid ay idoduplicate niya sa interphase during the S phase, the synthesis of the DNA. Ayan. Tingnan natin kasi ito in more diagrammatic. Ito kasi siya pinakita ko sa inyo. This is actual micrograph. Yung talagang picture under microscope. So, tingnan natin siya diagrammatically lang para makita natin yung buong cell. Yung chromosome, I should say. So, here, in the early prophase, you will see that the chromosomes are beginning to condense. Ayan. Nagko-condense na sila. Tapos, when we go to late prophase, makikita nyo that they are properly condensed now, showing the chromosomes with two sister chromatids. Ayan. So, in pro-metaphase, nakikita nyo yung chromosomes, may mga naka-attach na microtubules and spindle apparatus to each of the chromosome. Ready na silang hilain. No? Opposite pole, may dalawang pole tayo, the north pole and the south pole. At makikita mo that during the metaphase stage, ayan, they are properly aligned, guided by the spindle apparatus. Kada kinitukor ng bawat sister chromatid in preparation to, ano mangyayari sa anaphase? Yan, hinila ng spindle apparatus to the opposite pole. So, yung isang sister chromatid will go to the opposite and another sister chromatid will go to opposite. And now we have two daughter cells at the end of telophase. And siyempre, sa interphase, ganun din yan. No? Magkakaroon ng dalawang daughter cells that will undergo duplication of chromosomes during G1, G2, and the S phase. Okay. Tuloy-tuloy natin, ha? Siyempre, telophase will be followed by cytokinesis. Cytokinesis meaning division of the cytoplasm. So, tignan natin. In plant cell, what happens? No, it's like this. So, there is cell plate formation. Cytokinesis occurs by cell plate formation in plant cell. So, toward the end of the mitosis, vesicles gather at the middle. And then, yan, vesicles contain cementing materials and structural materials for a new primary cell wall. The plasma membranes of the vesicles fuse, sandwiching the contents. So, may hiwalay na, no? Mayroong kabilang pole at sa kabilang pole. So, to continue, a cell plate starts forming as membranes of the vesicles fuse. So, magkakahiwalay na sila ngayon. Primarily, nakasandwich siya. And this cell plate will elongate so that it will totally divide the cell into two daughter cells. So, this is what is happening among plants. Paano naman in animal cells? So, in animal cells, it goes like this. Now, so you have here a typical animal cell that undergoes cytoplasmic division. So, just beneath the plasma membrane, a band of microfilaments at the former spindle equator will form, and there will be a furrow that deepens until eventually these two will separate as daughter cells. So, itong two terms tandaan niyo. Pag plant cell, ang tawag natin ay cell plate. Pag animal cell, ang tawag natin ay cleavage furrow or cell furrow. Okay. My question, class. I have a question in here. Paano kaya? Sige lang, anyway. Makikita nyo yung answer agad kasi hindi ako naka-flash, naka-slideshow eh. The question is, if human has 23 pairs or 46 number of chromosomes, how many chromosomes will there be at each pole after anaphase? Diba nga, sa anaphase na stage, maghihiwalay. Ayan, diba? Ito yung anaphase na stage. Naghiwalay sila pero tag-isang sister chromatids lang. So, kung dito sa picture na to, 4 in each pole pareho lang. Pero kung human being, 46 pa rin yan doon sa isang pole, 46 pa rin sa other pole. Kasi ang nag-separate lang, sister chromatids. Characteristically, they are single-stranded. During the S phase of the interphase, magdo-duplicate yan uli in preparation of the next mitosis cycle. So, paano natin class nakokontrol itong cell division? So, the control of cell division by checkpoints. Very important, no? There is checkpoints in each part of the cycle. So, first, we have the cell growth checkpoint. This occurs towards the end of the G1. Checks whether the cell is big enough and has made the proper proteins for the synthesis phase. So if not, this goes to a resting period called the cell or the G0. Itong G0, bihira ito siya class matackle sa mga cell cycles. But in here, you just have to make sure that G0 happens when the cell is not ready to undergo the synthesis phase. The next checkpoint is what we call the DNA synthesis checkpoint. This occurs during the synthesis phase, or the S, checks whether DNA has been replicated correctly. If so, the cell continues on to mitosis. And then, the mitosis checkpoint occurs during the mitosis phase and checks whether mitosis is complete. If so, the cell cycle continues. The cell divides and the cycle repeats. This one is very important. Control of cell division. Two mechanisms that control cell division are the chemical signals such as the growth factors, the hormones, etc., and the availability of space. So, normal cells stop proliferating when they begin touching, or a phenomenon called contact inhibition. Di ba kasi nga, control ng cell division, availability ng space. So, kung yung space na dapat ma-occupy is na-meet na siya, makukontak pa rin. Siyempre kasi dikit-dikit o gano'n, diba? So, pag nagkaramdaman na katabing cell na niya, mag-stop na siya. Pero, sometimes, nangyayari dito class yung pag nawala yung normal na control nitong contact inhibition, dito nangyayari yung mga cancer cells. Cancer cells lack many of the normal controls and therefore divide wildly, which makes them dangerous for their host or to their host. So marami ng iba't ibang klase ng cancer. I will have a separate talk on the cancer as this is very special to me. No? Marami akong experiences dito that I would like to share with you. So this is all for the cell cycle. Do you have any question, class? Any question? Alright. Ang cell mall ko na klase. It's now your turn to ask question. Okay, since we're done, I would like you class to type your name na so that I will know that you are still present to the end while I am waiting for the